So what I'd like to do in this case study today is show you how a study that was originally designed and planned as a proof of concept study in a disease called Leber's Hereditary Optic Neuropathy, or LHON for short, transformed over time into a study which had a more pivotal type of character. And I would like to emphasize the reasons and the rationale why we had to change the study in the course of the 3 years that it lasted. And I would like to stress the importance and necessity to have regular interactions with health authorities to get their buy-in and support in order to allow these drastic changes to a study protocol happen, as I will going to show you. I should say that the outcome of the study, which I only will touch very briefly, has now formed the basis for a marketing authorization application that is currently under review by EMA's CHMP. But coming back to the disease itself, I imagine that very few people in the audience have ever heard about LHON. What is a very devastating form of blindness. It affects typically healthy young individuals, and a typical patient would be 30 years of age, would present with the doctor complaining about rapid vision loss. And the vision loss manifests in two aspects. It is color desaturation and confusion of color contrast perception in the red, green, and blue, yellow color domains, and I show you a little clip to illustrate that in a minute. But more devastating is the fact that the central scotoma develops. It's basically a black hole in the center of the vision, which rapidly evolves and expands, taking away the visual acuity of the patients within a matter of days or weeks. The outcome of the fate of the patient is devastating, because the patient will turn blind within a matter of weeks and remains blind for the rest of their life. Now, I have tried to mimic what a patient actually experiences. And this is shown in the next slide. I apologize, I hope that is working. So look at that slide. So first of all, color desaturation, confusion of the colors, and the central scotoma develops. It enlarges and may encompass the vast majority of the visual field. Basically, you're turning blind within a matter of weeks or months. Now, the disease is rare, but it still affects, the prevalence is about 2 in 100,000 in North America and in Europe. And as I said, a patient who turned blind has virtually no chance of spontaneous recovery. Now, the disease is very well understood on a molecular level. Illustrated here are the principal cause of the disease are 3 primary mitochondrial DNA mutations at the positions 11778, 14484, and 3460. These 3-point mutations, either of which is able to cause a conformational change in the complex 1 of the electron transport chain units in the mitochondria. And this has 2 devastating consequences. It causes a reactive oxygen species, very aggressive molecules, to form. These cell-damaging molecules cause further impaired function of the mitochondrial electron transport chain. And it causes a reduced generation of cellular energy, ATP. Now, this alone would not cause the blindness, but in conjunction with environmental factors such as exposure to certain anesthetics, toxins such as pesticides, or heavy smoking, or genetic predispositions over here, they come together, and then in a proportion of about 50% of carriers actually cause the blindness. The interesting aspect of this disease is that it exclusively affects the retinal ganglion cells of the eye. These are the cells that connect the retina to the brain. All the other cellular layers in the retina or the brain visual system are not affected. In fact, it's a very delicate and exclusive disease of the retinal ganglion cells. Now, this reduced ATP output and increased oxidative stress renders retinal ganglion cells dysfunctional. And this can happen over up to 5 years. But eventually, the cells die and are lost. Now, we are developing a molecule called idebenone, and we have a proprietary form of that. It's called catena, because we understand the mode of action of this molecule. What it does, it is a very powerful antioxidant. It goes into the cell and quenches reactive oxygen species. but it also can bypass the deficient complex 1 of the electron transport chain and shuttles electrons directly from the cytosol, which involves an enzymatic step, onto complex 3 of the electron transport chain, thereby bypassing the deficient complex 1. Now, we thought that this mode of action, which is well understood in the biochemical level and also in animal models, would be an ideal candidate for the treatment of Leber's hereditary optic neuropathy. And so about 4 years ago, we started to conduct and plan a clinical study in that disease. Now, the initial therapeutic goal that we had in mind was to come in early in the course of the disease and allow to prevent vision loss. So we wanted to test whether idebenone could prevent the vision loss that happens in these patients. That's illustrated here. Once again, a typical patient complains about vision loss in one eye, which can, over a matter of very few weeks, can progress to the level that the patient is basically blind on that eye. And then the second eye follows typically within the time frame of 3, 4, 5 months, often or sometimes also more quickly. And so the second eye is at risk of vision loss. So once a patient is characterized as a Leber's patient, first eye heavily affected, second eye about to be lost. So the therapeutic goal for that aspect was to prevent vision loss. So we designed the study, which we call the RODO study, and this stands for Rescue of Hereditary Optic Disease Outpatient Study. And actually went back to what was known at the time about the disease and went back to initiative taken by KOLs active. In fact, it was the first ever randomized controlled clinical study in that disease, which already tells you what we didn't know. We didn't know a lot about the medical history that we now understand 4 or 5 years later, but in the planning assumptions, very little knowledge was available because there was never a clinical study conducted before. So it was originally planned as an exploratory study with a treatment period of 9 months. And we had 2 centers, one center in Germany and one in UK, who wanted to participate and had patients. We planned to enroll 60 patients. And one of the key assumptions was, as we wanted to treat or look for prevention of vision loss, we wanted to enroll patients who had only been recently diagnosed. Patients who had had their diagnosis within 3 months from being enrolled. This was the idea. And we had a dose from previous experiments, and we proposed a 1-to-1 randomization in the study. Now, the endpoint that we selected at that time was we wanted to test what proportion of patients in both treatment groups would deteriorate significantly in their visual acuity. Now, this is illustrated here. Let's assume a newly diagnosed patient. Both eyes are somewhat affected, one eye more affected than the other. What you see here is the typical chart that is used to assess visual acuity. That's called an ETDRS chart. And what actually you measure is you count the number of letters that a patient can read, starting from the very small ones to increasingly larger size. Now, an important aspect that is important also for the regulators and also for the eventually for reimbursement is the fact that patients who reach what is called a logMAR unit of 1.0, they are classified as legally blind. LogMAR stands for logarithmic of the minimum angle of resolution. That's a numerical value for these letter charts here. Once you are reaching logMAR 1.0, you're not allowed to drive a vehicle or operate machinery. So this is a stringent definition that is in place in most of European countries. So we were assuming that patients who come in early into the study, we wanted to count the proportion of patients in whom the least affected eye would not deteriorate to this threshold of logMAR 1.0, prevention of vision loss. Now, in reality, we were faced that we started the study in open for enrollment in November 2006, and until September 2007, we had not a single patient enrolled. Despite the fact that we did all things you do, you had advertisements in journals, you were in congresses, and so forth. And in the period that we had no access to patients who were just diagnosed 3 months before, because the patients refused to enroll in a placebo-controlled study at a time when they learned about the devastating fate they were experiencing. They were too confused, they were too irritated, they didn't understand the fact that a placebo-controlled study would be needed to find out whether the drug would work or not. So we were forced to do something with the study. And so the study was amended, and we decided, with support of the investigators, to open it up for all comers. Basically, we said, we allow everybody in the study who has up to 5 years of disease history. So we changed the study radically. And this also changed the therapeutic goal, because opening up the study for patients up to 5 years into the disease would no longer mean that we could look at the prevention of vision loss, because we expected, from what was known at the time in natural history, that these patients already had lost their visual acuity. So we were turning now from prevention of vision loss to recovery of vision. We were interested whether patients who had lost their vision could come back and see better over time. Now, so the design that we came up with, the revised design, was we shortened the study from 9 months to 6 months. And this was only to motivate patients to come into the study. Still, it was a placebo-controlled trial. We added one more center in Canada and planned for a higher number of patients to be enrolled. But most importantly, we allowed patients in who had vision loss experienced up to 5 years prior to randomization. And we changed the randomization schedule from 1-to-1 to 2-to-1 in order to motivate patients to come forward to such a study. And you see, by doing that, and highlighted in red here, we changed dramatically key design elements of the clinical study. From the previous design to that one, it was a dramatic change. We morphed it from one study type to a completely different type of study. Now, that also brought about some changes in thinking of endpoints. We were thinking, based on the knowledge that was available at the time, that patients who would come in with a disease history of 5 years would be completely blind. That's illustrated in this little chart here. I show you a log-Mar scale, and here the charts to illustrate where we are on the scale. And I have illustrated here patients. Every patient are 2 dots, and this is both eyes of a patient. And we were assuming, based on the knowledge at the time, that patients with up to 5 years of history would have lost completely their visual acuity. They would be severely affected, even to the extent that they were no longer able to read any letters on such a chart. They would only be able to detect hand motion or count fingers in near distance from the face. That is what experts told us. That is what a typical patient after 5 years looked like, totally blind. Okay, then we said, if this is true, we pick the following endpoint. For these patients, the best recovery, visual acuity recovery, would be the clinically meaningful endpoint. And this was then selected as the primary endpoint of the study. Now, in reality, when the study started to recruit patients, we got phone calls from the investigators say, well, listen, we get patients in who have one eye very badly affected, but they have some still residual vision in the second eye. Now, this is again illustrated in this chart here. Again, patients, every patient has 2 eyes. So you have here patients, for instance, they are off chart. One eye is only able to detect hand motion, but the better eye is still able to detect counting fingers in near distance. Or even take those here in the right part. These are patients who have fairly good vision retained in one eye, but the second eye is already advanced to very bad visual acuity. This was not known. Nobody knew that patients after 5 years, still there are some out there who have completely different, we call it now discordant visual acuity in both eyes. Now we realized with this situation, our primary endpoint of best recovery is no longer valid. Because for these patients, the important aspect is that they should not lose that eye turning blind. So we had thought another endpoint would be more appropriate for this mixed population, and that is the best visual acuity, or a change in best visual acuity between the baseline and week 24. This is illustrated here. So if this new endpoint that we proposed would actually detect an improvement in this hypothetical patient 1 from baseline to week 24, because the visual acuity of this patient has improved from here to here. Or it would detect if a visual acuity in a patient worsens, illustrated in patient number 2, this patient happens to lose visual acuity, so this is one of those here, goes up to maybe counting fingers or even worse. So the best visual acuity, we realized, would have been the best endpoint for the study, but we didn't know when we started and select the primary endpoint. So we went for scientific advice. We met the CHMP and proposed and presented our dilemma. And I think it's interesting to learn that the CHMP realized that the primary endpoint in the study is not medically the most relevant for this heterogeneous population. So we have written recommendation from the CHMP that says that the company may wish to position the secondary endpoint that just proposed as main secondary endpoint to allow label claims to be made if positive results are seen. Remarkably, they did not say change your primary endpoint. They say pick your medically most relevant endpoint and call it main secondary endpoint. And they also said that this endpoint may be clinically more relevant than the primary endpoint and will be of special interest in the assessment. So here we have a situation where, in agreement with CHMP, the main secondary endpoint is the endpoint to look at, not the primary endpoint. Unusual situation. Now, in order to get further insight of this regulatory support, it's the following. From the recruitment rate, it became clear that the original design of the study was not, cannot be concluded, cannot be recruited in a reasonable time frame. So we had, in quick succession and before database lock, several meetings with the European competent authorities, both at the national level, MHRA, and at the EMA level. And we approached them for scientific advice and protocol assistance. And I think I have it somewhere on the slide. We had, within 12 or 14 months, we had 6 meetings with authorities in order to discuss the changes in the protocols we were entertaining. As I said already, the clinical relevance of the main secondary endpoint was endorsed by the CHMP, and it was positioned as the more medically relevant endpoint. And we also asked the authorities, as we realized that such a study, which already at this time run for 3 years, we cannot repeat in the near future, whether we should do an interim analysis to further test whether our statistical assumptions were correct. And we're clearly discouraged to do that. The authorities discouraged an interim analysis. They said we should go ahead and enroll as many patients as we can find, close the study and analyze it. They would be more willing to accept a good trend in efficacy in a completely clean study than deal with what they call, when we muddy the water by introducing an interim analysis. So that has been discussed, and I think in the last bullet it reads, they indicated the authorities that in case the trial results provide a statistically and clinically compelling demonstration of a positive risk-benefit, it may be possible to actually gain approval on this one study alone. Now, these are actually the data, very high-level summary, and just wanted to say that across multiple endpoints, without going into any details, looking at the main secondary endpoint here, best visual acuity in the ITT population and predefined and post-hoc populations, we saw a consistent pattern of idebenone being superior to placebo. But I have 2 summary slides I would like to show you, and that is, first of all, we again, as we heard today, faced enormous challenges in the lack of natural history data in preparing such a study. We introduced substantial changes to the key design elements of the study, changed inclusion, exclusion criteria, changed endpoints, and changed study objectives. And all these changes were made prior to study completion and in agreement with advice provided by competent authorities. And as I said before, overall we had 6 meetings with competent authorities to discuss these changes, and before we filed for marketing authorization application in July this year. Now, lessons learned. First of all, prospective planning of clinical or studies in orphan disease is challenging, and we have heard that multiple times this morning, and mainly due to the lack of natural history data. There are a possibility to radically change the scope, design elements, and even endpoints in clinical studies while maintaining the integrity following regulatory advice, provided that the study is fully blinded by the time you introduce the changes and you obey the recommendations given by the authorities. Regular meetings with competent authorities are possible and are highly recommended, and I think it's probably easier for the smaller companies to see them on a regular basis than for the big companies. And I should say that in conjunction with the regulatory framework currently in place with the EMA, such as the guidelines for clinical trials in small populations, this may allow, in specific circumstances, to optimize the study design for a single study to become more a pivotal type of study. And with this, I'd like to conclude my presentation. Thank you very much.